vlog, I'm going to show images of Canavan disease, which is also a rare leukodystrophy with a birth incidence of 1 in 100,000. But in Ashkenazi Jewish people, it is about 10 times as frequent, and 1 in 40 is a carrier of an SPA gene mutation that leads to Canavan disease. If I would ask a resident what he or she knows about Canavan, I hope the answer will be that it is a leukodystrophy with an increase in NAA. There are different forms of Canavan, and the most frequent is the infantile form that presents at the age of a few months. It has a very severe cause, and patients usually die by the age of 10. This 2-year-old girl presented with seizures, macrocephaly and hypotonia, and on imaging there was extensive hyperintensity of the white matter with sparing of the internal capsule and also hyperintensity in the thalami and globus pallidus, which is phylogenetically related to the thalamus. This is a 21-month-old boy with similar symptoms and a similar MRI, and on the lower images you can see that there is extensive involvement of the temporal lobe white matter and relative sparing of the cerebellar white matter. On spectroscopy, Canavan is the only disease that has an increase in NAA and elevated NAA choline and NAA creatine ratios. The juvenile form presents at the age where the children are in kindergarten and is much milder. And this girl had atypical imaging findings with hyperintensity in the caudate and putamen, thalami and infratentorial, but there was increased NAA on spectroscopy and also abnormal laboratory findings. The exact function of NAA is not known, so I'll share what we do know about NAA. It is synthesized in the mitochondria of the neuron, and there's normal NAA and NAA with glutamate coupled to it, and that's called NAAG or NAA glutamate. This gets transported in the axon, and when there is an electrical signal and depolarization of the neuron, there's also efflux of NAA and NAAG to the extracellular space. NAA glutamate binds to the glutamate receptor and inhibits the release of glutamate and also of other neurotransmitters such as GABA and dopamine. The NAA and NAAG are taken up from the extracellular space by oligodendrocytes and astrocytes, respectively. When we zoom in, and this tricellular cooperation is very important for the homeostasis of the white matter. And when we zoom in to the neuron and the oligodendrocyte, and there's reuptake of NAA by the oligodendrocyte, there's a catabolic pathway of NAA, and that's done by a protein, an enzyme called ASPA, and that's mutated in Canavan, and it's mainly present in the oligodendrocytes. And NAA in the oligodendrocytes is broken down into lipids that can be used for the formation of myelin, and an aspartate that serves as a supply for energy. And if you've seen previous vlogs, you might remember that in the one about hemimegalencephaly, I said that you could identify the abnormal hemisphere with seizures by the premature myelination, because myelination is driven by neural experience. And it sounded very logical, myelination driven by neural experience. And this depolarization and efflux of NAA and the breakdown of NAA and the oligodendrocyte into lipids, which is a building block of myelin, is a piece of the puzzle how the neuron and the oligodendrocyte communicate. So NAA is, how I imagine it, the way for the neuron to tell the oligodendrocyte, I'm active, I'm being depolarized, make or maintain my myelin. And if you look at the most recent classification of leukodystrophies, Canavan is classified as myelin vacuolization. The myelin vacuolization, interestingly, is not caused by the high NAA, which is shown in this experiment in mice from 2017. There were 3 groups of mice: a control group, a genetically normal group that was treated for 30 days with abnormal high levels of NAA, and a mouse model of Canavan where the mice were named NUR7 mice. And after this period, the mice were sacrificed. The controls, of course, had normal histopathology in the region of the third ventricle and the habenula. And in the hippocampal region, the Canavan-like mice had extensive cystic myelin degeneration in both the habenular region and in the hippocampus. And the mice treated with high levels of NAA similar to levels seen in Canavan disease remarkably did not have cystic degeneration. So there's not a linear correlation between the increase in NAA and the cystic degeneration in Canavan. There must be more to it. In the differential diagnosis of Canavan disease is another disease with macrocephaly, that's Alexander disease, which has an anterior white matter involvement and contrast enhancement. The white matter abnormalities in Canavan's disease do not enhance. And Alexander disease is a problem with the astrocyte. And another leukodystrophy with a problem with the astrocytes is Van der Knaap's disease, or megalencephalic leukoencephalopathy with subcortical cysts, that you see in this image. I've discussed 4 leukoencephalopathies now, so I think we do have an idea of the homeostasis and metabolism of the white matter. So in the next vlog I will switch to the gray matter of the basal ganglia and metabolic Lee disease.